We are talking about embryo transfer here, and we have a nice little video clip I want to show you before we wrap up and move into our next reproductive technology. So why don't you go ahead and take a look at this, and this will hopefully kind of better illustrate or explain how nuclear transfer works.
Somatic cell nuclear transfer cloning results in a genetic replica of a donor cell. In therapeutic cloning, this replica eliminates the rejection immune response in patients typical in traditional tissue and organ donation. In reproductive cloning, a genetic copy of the donor organism is the result. Two cells are involved in SCNT, the somatic or non-reproductive donor cell, and a reproductive host egg cell. First, the nucleus is removed from the egg cell to create an enucleated egg. The nucleus is then removed from the donor cell and transplanted into the enucleated egg. The egg may be cultured and developed into a blastocyst. Embryonic stem cells are extracted from the blastocyst for therapeutic cloning. Stem cells are treated with specific growth factors to produce a desired differentiated cell type, such as neurons or skin cells. Or the egg can be implanted into a surrogate mother's womb, where it gestates into a cloned organism in what is known as reproductive cloning.
So this briefly kind of explain how embryo transfer can work. Obviously, towards the end, they kind of hit on the head of what we were talking about previously with embryo transfer and how that works with cattle. And again, this is a very similar process to cloning. Let's go to the next slide. So feel free to pause on this diagram or take a look at it, but this does a nice job kind of explaining how the process works. So again, we started with superovulation, where we have more eggs that are being produced, and then we initiate that superovulation, we inseminate, we allow that flushing process to occur with our catheter, then we ensure that all of the animals are in sync, or they're synchronous, and then the actual embryo transfer occurs. And then 9 months later, the new calf is born. So embryo transfer, again, very popular, new coming trend here in, especially in the dairy and beef industries. Again, this diagram is a little bit harder to see and harder to read, but basically this explains the process a little bit more closely and compares embryo transfer to adult cell cloning. We will briefly talk about in vitro fertilization here too, because it's pretty short. In vitro fertilization is the process of fertilizing eggs in a petri dish and then injecting the eggs into the surrogate mother. So the diagram over the right briefly walks you through the steps. First, the egg is produced by hormone therapy. The eggs are retrieved from the ovary. The eggs and sperm are then combined in the petri dish to allow for fertilization. And then those fertilized eggs are then introduced into the uterus. We're going to stop there. I'm going to have you take a look at the strong silk brain break. It's actually pretty fascinating. I was in a biotechnology class in college, and they showed us this video, and it's pretty incredible what biotechnology can do to help make advances for animals and also for humans. So this is an example of one that you may find also interesting. So why don't we stop there and catch you back here in a few minutes for the next section of videos.